Hi, I'm Neil Greenspan. I'm a professor of pathology at Case Western Reserve University and senior editor at Pathogens and Immunity. And I'm here today with Erica Ollmann Saphire, president and chief executive officer of the La Jolla Institute for Immunology, to talk about her work on hemorrhagic fever viruses and SARS-CoV-2. And she'll be speaking about these topics at the Antibodies as Drugs meeting, Keystone Symposium, that is, and Dr. Ollmann Saphire will be giving the keynote address at that meeting.
So for a lot of viruses that have a membrane envelope, there's a key heavily glycosylated unstable protein on the surface whose job is to attach to your human cell and drive itself in. And because its job in life is to evade your immune system while springing into a much more stable structure to roll downhill in that energetic barrier that allows it to fuse into your cells, you're dealing with a heavily glycosylated, constantly mutating and changing, highly unstable molecule. And that has to be your target for your vaccine design, and that has to be your target for antibodies. Looking at the structure can tell you a lot of different things. It can tell you what surfaces seem to be key for the most potent activity. Do these antibodies directly block receptor binding? Is that their mechanism? Do they block fusion? Is that their mechanism? Do they bridge subunits together? How important are the glycans? Are the glycans a hindrance to limit antibody access, meaning in a next generation vaccine, you might limit those to improve how the original germline antibodies can bind and start that response going? Or do we need to leave those glycans in because they're key for recognition? A lot of potent antibodies might bridge some glycan to some molecule. So the structure tells you a lot about the roadmap of what the target might be and what modifications you might make in the antigen to better present the kinds of antibodies that look like they're most effective. And if what you want to achieve with your vaccine is broadly protective immunity, not strain-specific immunity, you can look at the kinds of antibodies that are able to recognize multiple related viruses or multiple strains of the same virus and see what kind of places they hit. And maybe you might want to find ways of better displaying those in the vaccine instead of the more immunogenic, but more strain-specific parts. So that structure is a really good way of understanding what you're making and why and what the end result is. But there's a lot beyond it. So for example, the reason we launched the large Ebola virus consortium is that we had done all the work of figuring out what is the structure of that surface antigen for Ebola and different viruses like it, and where do human antibodies bind that could achieve potent neutralization in vitro. But potent neutralization in cell culture didn't always translate to effective protection in a living thing. And so there are differences. It could be that the kind of antibodies that look fantastic in cell culture just have poor pharmacokinetics in the animal. It could be that their activity is easily recognized in the viro cells that are what you're using in your neutralization assay, but the antibody doesn't block activity in an infection of a different cell type that maybe uses a different receptor or has a different cleavage mechanism. So the antibody will effectively block in that one kind of cell, but in a living thing, the virus has found a backdoor. And so you need to combine scientists with a lot of different kinds of expertise. You need structure, you need glycan analysis, you need in vivo modeling, you need to understand what you can and can't interpret from the animal models. You need people that can measure PK, you need people that can decide, are these therapeutics even manufacturable? It might have something that you love so much at an academic level, but the protein won't express well enough, or it's just not bioavailable, it's not going to become an effective therapeutic.